Hello and good evening, everyone. So welcome to another live session on Edentu. So we are doing a series of sessions discussing the important MCQs from each subject. So today we'll be discussing mainly the topics from dental histology. So on my last live session, we had discussed about enamel, dentine, pulp, and cementum and everything. So in this session, we'll be mainly discussing on the salivary glands, oral mucous membrane, TMJ, and bone, and some histopathological technique questions also, okay, related to bone. Okay, so like I already told in my previous session, dental histology is like a fundamental subject. And for NEET exam, dental histology together with dental anatomy, you can expect a total of like 14 to 15 questions. And from dental histology alone, you can expect a total of like 7 to 8 questions. So today we'll be mainly discussing the MCQs from where the questions were like repeatedly asked. Okay, so we'll be discussing mainly that today. So if you ask me like what pattern of questions can be asked in dental histology, then I can tell you that you can expect like image-based questions. You can expect application or practical-based questions, and also you can expect like factual or concept-based one-liner questions also. So in this session, we'll be mainly focusing on image-based questions. Okay. The majority of the questions that I included in for this session is mainly image-based questions, and some one-liner questions were also included. Okay, so let's begin, doctors. So the first question is, the cell shown in the picture is found usually in the basal layer of the epithelium of oral mucosa. These cells are few in number, present adjacent to the unmyelinated nerve endings, and contain membrane-bound granules. Identify this cell. Okay, so this is the electron microscopy picture of this cell. Okay, so you have four options here. The options are melanocytes, lymphocytes, Merkel cells, and the Langerhans cells. Okay, all the non-keratinocytes. So if you look at the oral cavity, in the oral cavity, you have both the keratinized epithelium and the non-keratinized epithelium also. So likewise, we have the keratinocytes and the non-keratinocyte cell also. So these non-keratinocyte cells are also called as dendritic cells or the clear cells. Okay. So all the four options given here are of these non-keratinocyte cells that are present in the oral cavity. Okay. So they are asking you to identify which type of cell it is. Now, if you read the question, there'll be like a key point in every question that will take you directly to the answer. Now, the key point in this question is it is present adjacent to the unmyelinated nerve endings. Okay, so, and contain membrane-bound granules. Okay, so the cell which is present near the unmyelinated nerve endings and which has a role to play in the like the sensation is the mainly the option C, Merkel cells. Okay. So let's see what these Merkel cells are. So like I already mentioned, the cells in the oral cavity can be divided into two types. It can be like keratinocytes and non-keratinocytes. In non-keratinocytes, it includes the melanocytes, Langerhans cells, Merkel cells, and the inflammatory cells like the lymphocytes or the plasma cells or the mast cells. So what are these Merkel cells? Okay, so these Merkel cells are present both in the oral cavity and in the skin also. Okay, so this is one diagram showing the Merkel cells in the skin. Okay, so as you can see, they are present in the basal layer. Okay, so there are like 4 layers in the epithelium. So these Merkel cells, they will be mainly seen in the stratum basal. Okay, and if you look at the origin of these Merkel cells, they are mainly derived from the neural crest cells. And like I had already mentioned, since they are like present adjacent to the nerves or they are adjacent to the unmyelinated nerve fibers, what their main function is that they are like a sensitive receptor cell. Okay, they are a specialized neural pressure cell, and their main function is like a sensory cell they are acting. Okay, and like they are always present adjacent to the nerve tissue, and it is most commonly seen in our masticatory mucosa. Okay, like the gingiva. Okay, so these Merkel cells are most commonly seen in the masticatory mucosa like the gingiva, and compared to other non-keratinocytes like melanocytes, Langerhans, and all the other cells. These Merkel cells are non-dendritic. Okay, so these other cells like melanocytes and Langerhans, they have the dendritic processes extending from the cell body, but this Merkel cells alone, they don't have any dendritic processes and all. So they are the only non-dendritic, non-keratinocyte that is present in our oral mucosa. Now, if you look at the nucleus of the Merkel cell, okay, so this is the nucleus of the Merkel cell. You can see that it is somewhat like a roadlet shape, okay. It is a roadlet shaped and it has deep invaginations like this. Okay, so this is what is a characteristic appearance of the nucleus of the Merkel cell. It is roadlet shaped and it has deep invagination also. And another characteristic feature is that in the cytoplasm, you can see lot of granules like this. Okay, in the cytoplasm, you can see lot of granules like this. And usually these granules will be present near the nerve endings. Okay, so that is a characteristic feature of this Merkel cell. And it is like a little bit difficult to identify this Merkel cell with the normal hematoxylin and eosin stains. So we use something as a special stain. Okay, periodic acid shift stain. Okay, that is the PAS stain. Okay, with the help of this PAS stain, we can see this Merkel cells very clearly. Okay, so that's all about Merkel cells. Now, next question. What kind of secretion is shown in the histological picture of salivary gland below? Okay, is it apocrine or paracrine or merocrine or holocrine? Okay, so this is the picture, and you can see that it is already given that the cell gets pinched off in the secretion and the secretory products are stored in the cells like this. Okay, so a part of the cell gets pinched off in this secretion. So what kind of secretion is this? Is it apocrine or paracrine or merocrine or holocrine? Actually, it is option A, apocrine. Okay, so in our human body, we have like two type of glands. Okay, one is the endocrine glands and the other one is the exocrine glands. Okay, endocrine glands secrete hormones and exocrine glands secrete enzymes. Okay, so if you look at the endocrine glands, you can see that their secretory product, okay, their secretory product is directly released into the blood, and the blood will carry it to the all the organs. Okay, so it is like from the manufacturing unit, they are secreted directly into the blood, and the blood will carry it to the other organs. But exocrine glands are not like that. Exocrine glands will have a manufacturing unit and a transport unit also. So these exocrine glands are usually located at a site away from the site of action. Okay, so they will not be actually present at the site of action, but a little bit away from the site of action because they have a transport unit of their own. So from the manufacture unit, what they does is that the enzymes that are secreted will be secreted into the tubular system, and these tubes will carry the enzymes to the site of action. So that is the main way by which these enzymes act. So there are three main types of these exocrine secretions. Okay, so these exocrine secretions can again be divided into three types. First is merocrine, then the second one is apocrine, and the third one is holocrine. Okay, so let's see what this merocrine, apocrine, and holocrine is. Okay, so merocrine secretions are the most common type of secretion, and usually this merocrine secretion is done by these eccrine glands. And what happens in this merocrine secretion is that you can see here, this is an example of the merocrine secretion. There will be like membrane-bound vesicles inside each unit. Okay, so this is the single secretory unit, and these are the cells here. And inside every cell, you can see like vesicle-bound structures like this. Okay, so these vesicle-bound structures contains the secretory product. So what they does is that they will go to the apical portion of this cell, and then they will release this vesicle outside. Okay, so from the vesicle, this secretory product is released to outside. Now, this secretion will go into the gut. Okay, such type of secretion is called as the merocrine secretion. Okay, the main examples are sweat glands, salivary glands, tear glands, and the goblet cells in the respiratory, digestive tract, and the intestinal glands are all examples of this merocrine type of secretion. Okay, now another type of secretion that is given in the question here is the apocrine secretion. Okay, this is an example of a apocrine secretion. You can see This is a secretory unit, and these are the cells, individual cells here. Now, what happens in this apocrine secretion is that the secretory product will come and like concentrate near the apical portion of the cells here. Okay, you can see that this secretory product will get come and gets concentrated in the apical portion here. And in apocrine secretion, what happens is that this apical portion will get pinched off. Okay, so the apical portion will get pinched off and will get released into the ductal area. Okay, so that you can see here, the pinched off portion of the cell in the secretion. Okay, so the whole apical portion will get pinched off and it will be secreted into the duct. Okay, likewise, the enzymes will be going. Okay, so the main example of this apocrine type of secretion is the mammary glands. Now, next is the holocrine type of secretion. Okay, like the name suggests, holocrine means the whole cell will get destroyed during the secretion process. Okay, as you can see here, the whole cell gets shed into the duct and it will be replaced by another underlying cell. Okay, so the whole cell dies and releases its secretory product into the duct. So it is called as the holocrine. So holocrine means the whole cell will get destroyed and release its contents. Apocrine means only the apical portion of the cell gets destroyed. Merocrine means the secretory product will be contained in a vesicles, and only the vesicles will be released. Okay, now one example of this holocrine type of secretion is the sebaceous glands. Now, in the question, there was one more option called the paracrine signaling. Okay, so let's see what this paracrine signaling is. Paracrine signaling is localized form of signaling, like between two cells. If you want to like communicate something, then they use this paracrine signaling. Okay, so in paracrine signaling, again, they have like small structures like this, and which contains the chemical molecules. So these chemical molecules will be released outside, and if the adjacent cell have the receptors to identify these chemicals, then they will identify it and will cause the essential changes in the other cell. Okay, so it will be like a localized form of signaling to the adjacent cell only. Okay, so this type of signaling is called as the paracrine signal. Okay, now next question is, the given histological picture is of. Okay, so this histological picture, what does this histological picture indicate? Okay, you all the 4 papillae are given. Now choose the correct papillae. Okay, so is it filiform papillae, or is it fungiform papillae, or is it foliate papillae, or is it circumvallate papillae? Okay, answer is option D, circumvallate papillae. Okay, I'll tell you why is circumvallate papillae. So the papillae of the tongue can be divided into 4 types, as you all know. There are like filiform papillae, fungiform papillae, foliate papillae, and the circumvallate papillae. And filiform papillae, they are the most abundant form of papillae, and they are conical in shape, and they have no taste buds. Okay, filiform papillae have no taste buds, and they are present in front of the sulcus terminalis. Okay, then at the tip of the tongue, you can see the fungiform papillae, usually mushroom shaped, and they have taste buds. Okay, except filiform papillae, all other papillae have the taste buds. Okay, so next is the circumvallate papillae, which is the largest papillae. They are like 10 to 12 in number and present in front of this sulcus terminalis. Okay, these are the circumvallate papillae. And then you have the foliate papillae, which looks like the leaf ledges, and they are present on the margins of the tongue near the sulcus terminalis, and they also have the taste buds. Okay, so if you look at the histology of all these papillae, you can see that this is the histology of the filiform papillae. Okay, now one characteristic finding in this filiform papillae histological picture is the flame-shaped epithelium. Okay, you can see this flame-shaped epithelium like this. Okay, this is keratinized epithelium, and it forms like a flame-shaped areas in the topmost region. Okay, so this flame-shaped keratinized epithelium is a characteristic feature of this filiform papillae, and mainly usually the keratinization will be parakeratinized, and then you have the underlying connective tissue also. Now, if you look at the fungiform papillae, in the fungiform papillae, it will be usually like the the keratinization will not be seen. It will be usually non-keratinized, and lot of like blood vessels will be seen in the underlying connective tissue also. That's why it appears red in color. Okay, so it will be non-keratinized and lot of blood vessels in the underlying connective tissue. Okay. Now the one in question that is given here is the circumvallate papillae. Okay. Now one easy way to identify the circumvallate papillae is that this in this circumvallate papillae you will have these things. Okay, like there, like a crypt will be seen. Okay, between two papillae like region, papillar projections, you can see a and the scripts will be non-keratinized. Okay, that is one feature of the circumvallate papillae. The topmost layer will be keratinized. This topmost layer, okay, this topmost layer will be keratinized, whereas the scripts, you know, this invaginations or the crypts you can call, the scripts will be usually non-keratinized. So it is like a mixture of both the epitheliums. Okay, you can see both the keratinized epithelium and the non-keratinized epithelium within this one papillae itself. Okay, so that is one of the main characteristic feature of this circumvallate papillae. Okay, and underlying, you can see the von Ebner's gland and the skeletal tissue and the connective tissue. Okay, and in these crypts, you will be able to see the taste buds also. Okay, so that's one characteristic feature of this circumvallate papillae. Now, next question. Identify the structure marked with arrow in the histological picture given below. Okay. So it is evident that it is a salivary gland picture, and these are mucous acini, and these are like these cells, they look like they hug the mucous acini like this. Okay, so let's see what this is. Is it serous demileunes, or is it mucous acini, or is it myoepithelial cells, or striated duct? Okay. Answer is, it is option A, serous demileunes. Okay. So this is a histological picture of a mixed salivary gland. Okay. Mixed salivary glands include either the submandibular gland or the sublingual gland. Both are mixed salivary glands, but in the submandibular gland, the serous secretions will be more, whereas in the sublingual gland, mucous secretions will be more. Okay. So these are both mixed salivary glands, and as you can see, these are the serous acini, the darkly stained and blue color, the serous acini, and these lightly stained areas, lightly stained acini, they are the mucous acini with the nucleus pushed towards the basement membrane, and nucleus is usually flattened also. Okay, so these are the mucous acini, and these cells that are seen here, what are they? Was the question, in the question. So these cells are nothing but they are the serous gemmules that are seen in the mixed salivary glands. Okay, they will be like seen hugging, like the hugging the mucous acini. Okay, so demileunes means like half crescent shaped. Okay, so these things will be like half crescent shapes only. Now these serous demileunes are also called as crescents of Giannouchi, and it is also called as the demileunes of Heidenhain. Okay, now what are these serous demileunes? Okay, we can see the serous demileunes only in the mixed salivary glands, and at first it was thought that it was the serous cells that are seen in a mixed salivary gland. Okay, but later more and more investigations were done, and we instead of like doing the normal histology technique, what we did was we did a frozen section also. So when we did a frozen section, these serous demileunes were not there. Okay, so this is the normal histological H&E section picture where you can see the serous demileunes here. But in this is the picture of the frozen section done, where there was no serous demileunes. The cells were like in between the mucous cells itself. Okay, the serous cells were seen in between the mucous cells itself, and they were not like seen like above the mucous acini like that. Okay, so the pathologists, they concluded that these serous demileunes are nothing but they are just an artifact. Okay, while doing the tissue processing. Okay, tissue processing includes like lot of techniques, and lot of chemicals are used, and lot of these things are used. So what they like concluded what this, these serous demileunes are nothing but they are an artifact because of some tissue processing errors or something like that. Because of that only these serous demileunes are present, and it is not an actual structure that is present in the histology of the salivary glands. Okay, so this is actually how a mixed salivary gland histopathology will look like. Okay, intermixing of the both the serous and the mucous cells. So I hope I'm clear about the serous demilures. Now coming to the next question, identify the structure in the given image. Okay, this structure, okay, behind every permanent teeth, okay, this small canal-like structures, what are they? Okay, is it gubernacular canals, or is it an incisive foramen, or is it canals of Hirschfeld and Zucker-Candle, or is it Walkman's canal? Okay, actually it is option A, gubernacular canals. Okay, so what are these gubernacular canals? Okay, so these gubernacular canals are nothing but these gubernacular canals are usually seen in the lingual side of the permanent teeth, which has deciduous predecessors. Okay, like it is present only in those permanent teeth which has the deciduous predecessors. Okay, like the incisors and the canines and the premolars only. Okay, so what this gubernacular canal contains is, this gubernacular canal contains a small fibrous tissue which is called as the gubernacular cord. Okay, or it is also called as gubernacular dentis. Okay, so this is the gubernacular cord or gubernacular dentis. Okay, and it is present inside a bony canal which is called as the gubernacular canal. Okay, this is present in the lingual side or the palatal side. Look, you can see this. This is present in the lingual side or the palatal side of the deciduous teeth. Okay, so what the main function of this gubernacular dentis is that it acts as a guide for eruption of the permanent teeth. Like it is like a guiding force for the eruption of the permanent teeth. Okay, it actually makes way for the eruption of the permanent teeth. Okay, so this gubernacular cord is nothing but it is an anatomical structure that connects the dental follicle of the permanent tooth from the dental follicle to the overlying gingiva. It is extending and it helps in the eruption of the permanent tooth. Okay, so this gubernacular canal or tract means it is the surrounding bony canal, and the main function is the eruption. Okay, so moving on to the next question, which of the following is used as a decalcifying agent? Okay, is it alcohol or formalin or nitric acid or xylene? Okay, so tissues like teeth and bone, we can't just cut these tissues as such and look it under the microscope. Okay, it will be one thing. It will bone and teeth are like very hard, so it will be very difficult to cut them and look it under the microscope. Okay, so what we does for this tissues like bone and teeth are that we do a procedure called decalcification. Okay, decalcification, like the name suggests, it means removing the calcium from the teeth and bone. Okay, so once the calcium is removed, these teeth and bone, they will be like very soft, and then these structures will be easy to cut and it will be easy to stain them and it will be easy to look them under the microscope. Okay, so this procedure is called the decalcification, and the agent that we use for this decalcification is called as the decalcifying agent. Okay, so according to these four options given, which is the decalcifying agent? Okay, usually we use acids as decalcifying agent. Okay, so let's see. So for any tissue or like mostly for studying under the microscope, there are like four types of preparation that we usually use for studying a tissue under the microscope. One is the formalin fixed paraffin embedded sections. Okay, that is what like you had seen in the previous slides, like the circumvallate papillae and all the other normally stained tissues that are all. And the next we can do is this decalcified paraffin embedded section. Okay, this decalcification is mainly done for the hard tissues like the teeth and the bone. And then you have the ground sections also. Ground section means we are just reducing the tooth by rubbing it. Okay, so that is the ground section. And next we have the frozen section also. Okay, that's a little bit advanced. Okay, so Decalcification means it is the process of removal of the calcium from the tissue. And during this decalcification procedure, usually enamel will be completely lost. Okay, enamel will be completely lost because if you look at the composition of enamel, enamel is mainly composed of 96% inorganic substance. Okay, so while decalcification procedure, like the whole of the enamel will be lost and will not be able to see much of enamel in the decalcification process. So if you want to study some enamel defects and if you want to study the structure of the enamel, then decalcification is not the recommended procedure. The recommended procedure is always ground section for studying the enamel. Okay. But in case of like dentine, cementum and bone and all, you can do the decalcification procedure also. So what we do for in this decalcification procedure is first, if it is a tooth, okay, we are mainly concentrating on the tooth only. If it's a tooth, the tooth is extracted and a hole is made on the tooth and fixation is done. Okay, usually we does fixation for every tissue, that is before the tissue processing. So the fixation is done with the help of 10% neutral buffered formalin, and then the tooth is kept in 5% nitric acid. Okay, this acids are usually the agents that we use for decalcification. Usually used is the 5% nitric acid, and the what we do is that we will keep the tooth under in this nitric acid and we will daily change it. Okay, change the acid. Now, like after like 5 to 6 days or like 1 week, what we can see is that this once the calcium, the whole calcium is lost, this tissue will become very soft. Okay, so how can we like test whether the tissue has completed its decalcification processes? We can take a pin and try to pierce it. Okay, if the pin goes through the entire length of the tissue, then we can confirm that okay, the tissue has been decalcified and now we can use it for the further procedure. Okay, so once this pin test is satisfied, then what you can do is you can thoroughly wash this tissue under the running water, and then usual processing steps are done, like the clearing and the cutting and all the usual processing steps are done, and finally we will get a decalcified picture after staining and all. If you look at the microscope, we can get a decalcified tissue like this. Okay, here it is the dentine that is mainly seen. Okay, so that's about the decalcification procedure. Now the next question is stratum germinativum includes. Okay, this is recently asked NEET MDS question. The stratum germinativum means it includes stratum basal and parabasal spinous layer. Okay, so let's see what this stratum germinativum is. Okay, so if you look at a keratinized epithelium, you can see that there are 4 layers. Okay, the basement layer is the stratum basal, then above that you have the stratum spinosum, then above that you have the stratum granulosum, and then above that you have the stratum corneum. Okay, so these are the 4 layers that are present in the keratinized epithelium. Okay, so what is the stratum germinativum? Stratum germinativum means this basal layer. Okay, this is the basal layer here. Okay, this basal layer is composed of tall columnar cells and parabasal spinous layer. Okay, parabasal spinous layer means these layers, like 2 to 3 layers just above the basal layer. Okay, they are called as the parabasal layers. Okay, they are included in the stratum spinosum, but since they are just above the stratum basal, they are called as the parabasal spinous layer. Okay, so the stratum basal together with this parabasal spinous layer, they are called as the stratum germinativum. Okay, why this stratum germinativum we are calling that? Because it has high mitotic activity. Okay, all these stratum germinativum together, they have the high mitotic activity and it continuously produces new keratinocytes. Okay, because of its continuous proliferative nature, it is called as the stratum germinativum. Now, next question is identify the marked structure in the image given below. Okay, so this is cells with their junctions. Okay, so these are the junctions here, these are the junctions here, and there is junctions here also. So they are asking us to identify what this junction is called. Okay, is it desmosomes? Is it hemidesmosomes? Or is it called tight junction? Or is it called gap junction? Okay, answer is it is called as hemidesmosomes. So if you look at cell junctions, okay, cell junctions means there are two cells, and there is a junction between the two cells. These junctions are mainly for anchoring purposes, and they are also for the communication purposes. Okay, so these junctions can be divided into three types. You have the occluding junctions, anchoring junctions, and the communicating junctions. In the occluding junctions, it will be tight junctions. There will be no communication between the two types of cells. Okay, they are called as occluding junction. Then there are anchoring junctions. Okay, in the anchoring junction, there will be like two filaments. One is the actin filament, and the other one is the intermediate filament. Okay, communicating junctions mean to communicate between the two cells. There will be gaps also in between. Okay, so in the actin filament attachment sites, you have the cell to cell junctions, which are called as the adherence junction, and the cell to matrix junction. Okay, they are called as the focal adhesions. Okay. And in the intermediate filament attachment sites, you have something called as cell-to-cell junction. It is called as the desmosomes, and the cell-to-matrix junction, which is called as the hemidesmosomes. Okay, I will show the pictures so that it will be more clear for you. So this is an example of tight junction, no communication between the two cells. This is an example of adherence junction, like mainly for anchoring and those purposes. And this is the cell-to-cell junction. Okay, this is an example of the cell-to-cell junctions. The cell-to-cell junctions are called as the desmosomes, and this is the gap junction where there will be communication between the two cells. And from the cell to the basement membrane, we use something called as this hemidesmosomes. Okay, these hemidesmosomes are the anchoring units between the cell and the underlying basement membrane. So you should know the difference between the desmosomes and the hemidesmosomes. Desmosomes means between two cells. Hemidesmosomes means between the cell and the basement membrane. Okay, so you must be aware of that. Now, next question. Identify the marked structure in the bone adjacent to the periodontal ligament in the image given below. Okay, so this bone. Okay, the bone that is seen near the periodontal ligament. What is this bone called? Okay, is it lamina dura or bundle bone or lamina propria or lamina densa? Okay, actually it is called as bundle bone. Okay, now let me tell you about the parts of the alveolar bone. Okay, the bone that supports the tooth is called as the alveolar bone or the alveolar process, and under the alveolar process you have the basal bone. Okay, so there is the basal bone in the underneath region, and then the bone that supports the tooth, that is called as the alveolar process or the alveolar bone. Okay, so this alveolar bone is also called as pars alveolaris, and this alveolar bone is again divided into two. One is alveolar bone proper, or it is called as the cribriform plate, and the other one is the supporting alveolar bone. Okay, so this alveolar bone proper is again divided into two. One is the bundle bone and the lamellated bone. Okay, so the bundle bone means the bone, the site of attachment of periodontal fibers to the bone is known as the bundle bone. Okay, so to this bundle bone only, the periodontal fibers will get attached. Okay, and those fibers are called as the Sharpie's fibers. Okay, remember the name? Sharpie's fibers will get attached to the bundle bone. Okay, you can see a lot of Sharpie's fibers if you look at the histological section of this bundle bone. Okay, and usually these Sharpie's fibers will be attached at a oblique or the right angled areas. Okay, so this is the bundle bone that is present just adjacent to the periodontal ligament, where you will get you can see the attachment of the periodontal fibers. Okay, then you have the lamellated bone. Lamellated bone means the bone that is adjacent to the bundle bone. It is like a thin plate-like structure which is arranged parallel to each other. Okay, that is about the lamellated bone. Now moving on to the next question. The structure A shown in the following image is, okay, so what is this structure A? Okay, is it osteon, or is it osteoblast, or is it osteocyte, or is it osteoclast? Okay, it is actually osteon. Osteon is actually a single functional unit of the bone. Okay, so you can see this is the cross section of the compact bone, and you can see it will be like concentric circles will be there. Okay, this shows the like the periodic deposition of the bone over time. Okay, these concentric circles, they show the periodic deposition over a period of time. And all these, there will be like cells that are seen in the blue color here. These cells are called as the osteocytes. Okay, they are the osteocytes that are seen in the bone, which are the bone cells. Okay. Now we have in the option, we have also seen osteoblast, which are the bone forming cells, and we have seen osteoclast, which are the bone resorbing cells. Okay, here, if you look at the normal cross section of a compact bone, you can see these concentric rings like that, and there will be cells in the concentric rings also, which are called as the osteocytes. Okay, now if you take one concentric ring alone and concentrate on it, you can see that in the central, that single unit is called as the osteon, and at the center you can see something called as the Haversian canal. Okay, so this Haversian canal together with this concentric lamellae, they form the osteon or the Haversian system. Okay, so this is a single osteon, this is the Haversian canal, this is the concentric lamellae, and this is the osteocyte in the lacunum. Okay, so this mainly what the function of this osteon is, it encloses the vessels and nerves, okay, that is supplying the bone. Okay, it mainly encloses the vessels and nerves that is supplying the bone. Now, moving on to the next question, the temporomandibular joint is classified as which of the following types of joint found in the human body? Is it ball and socket joint, or is it gliding only joint, or hinge only joint, or gangliomo-arthroidal joint? Okay, so the correct answer is option D, gangliomo-arthroidal joint. So all the joints in our body can be divided into three types. First one is the fibrous or the immovable joints, and then you have the cartilaginous or the partially movable joints, and then you have the synovial or the freely movable joints. Okay, so this TMJ is actually a synovial joint, but under the synovial joint we can classify it as gangliomo-arthroidal joint also, because it has a ganglimus. Ganglimus is nothing but it is a freely moving joint in which like the bones are articulated to allow extensive movement in one plane. Okay, so such type of joints is called as the ganglimus, and the other one is the arthroidea. Arthroidea means sliding joints. It is a hollow area onto which the head of the other bone joints. Okay, so since both ganglimus and arthroidea are arthroidal, it is both ganglimus and arthroidal, then we can call it as the gangliomo-arthroidal joint. Okay, so remember the name. TMJ is a synovial joint and it is also a gangliomo-arthroidal joint. Now, moving on to the next question. Identify the curve shown in the image given below. Okay, so which is this curve? Okay, is it curve of Spee or curve of Munson or curve of Williams or curve of Wilson? Answer is option D, curve of Wilson. Okay, so let's see what this curve of Wilson is. Curve of Wilson is nothing but it is a mediolateral curve. Okay, it is a mediolateral curve that contacts the buccal and lingual cusp tips of each side of the arch. Okay, you can see in the picture here, this is a mediolateral curve that is joining the buccal and lingual cusp tip of the each side of the arch. Okay, why this Wilson curve is there? Because it is due to the buccal and lingual inclination of the maxillary and mandibular posteriors. Okay, so our lower posteriors, they will be a little bit lingually inclined, and our upper posteriors, maxillary posteriors, they will be a little bit buccally inclined. Okay, so because of the lingual inclination of the lower posterior, what happens is that the lingual cusp will be placed at a little bit lower position when compared to the buccal cusp in the mandibular arch. And if you see the maxillary arch, you can see that the maxillary posteriors will be a little bit buccally placed, so that their buccal cusp are usually higher than that of the lingual cusp. Okay, so in the, if you look at this Munson this Wilson curve can be drawn in the both the mandibular arch and in the maxillary arch. In the mandibular arch, you will usually have a concave shape, whereas in the maxillary arch, you will usually see a convex shape. Okay, so that is about the curve of Wilson. Now, this curve of Wilson is usually drawn like usually seen like in right angles to the curve of Spee. Okay, curve of Spee will be joining the buccal cusp tips of all the posteriors. So it will be, so if you can see that it will be usually seen at right angles to the curve of Spee. So that's all for today. If you have like any question, you can put it under the chat box under this video. We'll definitely answer it. And we'll be again coming with more live sessions. So in case of any doubts or in case of any difficulty, if you're finding in learning for this dental histology subject for your NEET MDS exam, you can definitely put your comments under this video. We will definitely answer it. Okay, so that's all for today. All the best for your preparations. Do well. Good night.